Chronic granulomatous disease is a disease that affects the immune system by hurting phagocytes. Phagocytes engulf and destroy foreign particles that could be harmful. The respiration burst pathway is a pathway that creates many superoxide ions and other molecules, such as hydrogen peroxide, that helps kill bacteria. This pathway is catalyzed by NADPH oxidase. Chronic granulomatous disease occurs when one of the genes that code for this protein is mutated. Because the protein cannot catalyze a respiratory burst, phagocytes are unable to destroy harmful particles. The mutation that we'll be talking about today is a mutation in the GBBE gene, which codes for a subunit of the NADPH oxidase. The gene is on chromosome X at position P21.1. The specific mutation we'll be targeting is a insertion mutation where bases 286 to 290 are repeated. PCR will be used to create more copies of this gene for study. To cut the DNA strand, we will be using the restriction enzyme TSPR1. On the screen, the N refers to any base, and S refers to either guanine or cytosine. We will also use the restriction enzyme PLE1. The restriction enzyme cuts will occur at position 252 and 403 on the gene. This is a model of how the DNA is being cut by the restriction enzymes. The cut sequence has a length of 150 base pairs. The primers I will be using for PCR are highlighted in orange on the screen. The target sequence will have a length of 40 base pairs. One way to treat this disease is by using non-homologous end-joining CRISPR, which can help cut out the extra base pairs. For this mutation, we will target the PAM sequence AGG, which is already in the mutated region. The mutation will be cut between the G and the G, 3 bases upstream from the AGG sequence. Due to the ligation process of the cell, the AGGAA sequence will eventually be removed, and thus the mutation will be fixed. We can use an adeno-associated virus to act as our vector for CRISPR. We will put the CRISPR-Cas9 and the guide RNA into the virus, and they will allow us to edit the DNA within the human body. Another possible cure to this disease is ex vivo gene therapy. Ex vivo gene therapy is better than in vivo in this instance because this disease affects the bloodstream. Because the host needs a way to keep producing the new and fixed protein, we will have to use a retrovirus so that it can integrate the working DNA into the cell's chromosome so it can replicate further. The hematopoietic stem cells will first be taken out of the patient's body. Then, a retrovirus with the correct code for the protein will integrate its DNA into the cell's chromosome. Finally, the cells will be injected back into the patient where they can replicate and produce the correct protein. This gel electrophoresis demonstrates polymorphism by showing how the mutated gene is different from the normal gene, because one piece of the mutated gene has a length of 150 base pairs, while the normal gene has one piece has a length of 145 base pairs.